This lecture is over Chapter 19. It covers the kidneys and the urinary system. I would refer you to your Chapter 19 study guide to see the learning objectives of which you'll be responsible. So there's 6 main functions of the kidney. The first function is going to be the regulation of extracellular fluid, volume, and blood pressure. The kidneys, by moving solutes in and out of the blood, can control the amount of water that is in the blood. And as we learned from our previous discussions over the cardiovascular system, an increase in water in our ECF will increase blood volume. An increase in blood volume can result in a higher blood pressure. So in addition to the extracellular fluid volume, we also regulate the osmolarity, being the number of solutes per liter of water. This leads us into the third function, which is the maintenance of ion balance. So the kidneys ensure that we have the proper amount of sodium, potassium, and calcium in our ECF to maintain the appropriate membrane dynamics needed for cellular functioning. Kidneys also aid in pH regulation. Now, this isn't as rapid as, say, in our lungs. The pH regulation of most of our blood is accomplished through CO2 and bicarbonate retention in the plasma of our blood. However, the kidneys also play a role in this pH regulation. The fifth function of the kidney is going to be the excretion of cellular wastes. And finally, the last function of the kidney is going to be the production of a handful of hormones, one of which is erythropoietin. That is a protein necessary. It stimulates red blood cell synthesis. We also produce the hormone renin in the kidneys, and this regulates some of the hormones that are involved in sodium balance and the blood pressure homeostasis. Now, if we look at the kidneys themselves in relation to the rest of the body, you will see that they're located in our abdomen, posterior, and that within the kidney themselves, there's these 2 main tissue types. We have the cortex, which is on the outside, and the medulla. And the functional unit of the kidney, the part that we'll be spending most of the time talking about, is called the nephron. So the nephron exists both in the cortex, and there's also regions that exist within the medulla. And it's here within each of these nephrons that are interspersed all throughout the kidney that filtration takes place. A couple points that you'll be responsible for. So while I don't test on a lot of anatomy, we can't talk about the nephron and filtration without understanding a few aspects and a few anatomical parts of the nephron. So the first is what we call the Bowman's capsule. So this is the part of the nephron that surrounds our capillary bed. So we move from the Bowman's capsule into the proximal tubule. Okay, then we will move into the descending limb of the loop of Henle. We'll have the ascending loop, move into the distal tubule, and finally into the collecting duct that leads to the bladder. Now, we spoke of the Bowman's capsule. That's the part of the lumen of the nephron. Associated with that Bowman's capsule are the capillary beds that run through that. We call that the glomerulus. Okay, so the glomerulus is the capillary. The Bowman's capsule is the nephron's lumen. And if we look at function of the nephron and the kidney as a whole, there are 4 processes that occur within the nephron. We have filtration, which is going to be the movement of blood from the glomerulus into the lumen of the nephron. We have reabsorption, which will be movement of fluid or solutes from the lumen back into the blood. We have secretion that occurs from blood elsewhere in the capillaries back into the lumen, and finally excretion, in which we move fluid and solutes from the lumen to outside of the body via the bladder. So just to recap, so filtration only takes place within the Bowman's capsule. Okay, sometimes we refer to this, both the glomerulus and the Bowman's capsule, as the renal corpuscle. But this is where all of our filtration will occur. Reabsorption can occur in the proximal tubules, the ascending, descending limb, distal tubule, and the collecting duct. Whereas secretion, it doesn't happen in the loops of Henle, but it can happen in the proximal tubule, distal tubule, and collecting duct. And excretion only occurs via the bladder. When we speak of filtration, it's important for us to recognize that if we look at an individual nephron, about 20% of all of the blood that runs through the afferent arteriole into the glomerulus is going to be initially filtered into the Bowman's capsule. So of that 20% that gets filtered in, only 1% of that volume will actually be excreted. So the other 19% is reabsorbed. So on a given day, let's say you knew that somebody was filtering roughly 120 milliliters of plasma every minute, if you could calculate then what the renal daily plasma flow would be of how much is moving through in a given day and how much ultimately gets filtered out. Recognizing that 99% of all of that filtrate is going to be retained. Don't worry, you won't be asked to calculate this on an exam, but I put this up there to show kind of what's going on, right? That we have a large percentage that ultimately gets filtered, but most will be reabsorbed. Not most, 99% of it will be reabsorbed. So how does this filtration occur? Well, to understand filtration, we have to understand that this is a process similar to what we were talking about with in respiration and cardiovascular, that's being driven by some hydrostatic pressures and bulk flow. Often when we speak of filtration, we'll talk about something called GFR, which stands for the glomerular filtration rate. So at what amount of blood is being pushed through the glomerular capillary into the lumen of the Bowman's capsule. And what influences this is going to be two factors. First is going to be, what is the net filtration pressure? What type of pressure is this blood under that pushes this into the lumen? And there's a couple of pressures that come into play. We'll talk about those on the next slide, but they're hydrostatic, colloid, and fluid pressure. The other aspect that's going to dictate what our filtration rate is what we call the filtration coefficient. And that is established by the surface area found within the capillary and the permeability between the capillary and the Bowman's capsule. And what you'll notice is where we have these capillaries come in, we have these specialized cells that are called podocytes. And these podocytes have these little foot processes that serve as that interface between the capillary and the Bowman's capsule. And depending on how tight these foot processes are, we could increase or decrease that filtration coefficient. Let's talk about these pressures. Okay, let's leave the filtration coefficient to the side and let's just talk about the pressure that's forcing this filtration. So I mentioned that there's three pressures. The first and greatest pressure is the hydrostatic pressure found in our arteriole. So blood coming in from the afferent renal arteriole is going to be under a hydrostatic pressure. That pressure is going to want to push it out from areas of high pressure to low pressure. And inside the lumen we have a lower pressure. Now, working against that hydrostatic pressure are two additional pressures. One is called the fluid pressure. Okay, this fluid pressure is the pressure found within the fluid in the lumen capsule. Now, it's not as great as the hydrostatic pressure. It's generally fairly low, but that's going to oppose that hydrostatic pressure to some degree. The second pressure is what we call colloid osmotic pressure. And this pressure is built due to the proteins that we find in our plasma, but not in the lumen. So blood proteins do not move into the lumen. They are not filtered. And so because of that, there's a pressure differential that's created by that plasma that opposes the hydrostatic pressure. So if we took all these into account, we'd get a net filtration pressure, which is generally always going to be leading towards filtration. Now, the body is incredibly efficient at regulating GFR, that glomerular filtration rate. And it's regulated primarily by the hydrostatic pressure found within the glomerulus. And so if we look at an average person, what we see is, at very low blood pressures, that GFR is going to be low. And then it increases on a linear scale until we reach about normal blood pressure at about 80 millimeters mercury. And between 80 and roughly 180, we have the same glomerular filtration rate, which is about 180 liters a day is what we filter out. So 180 liters per day get filtered. Again, 99% of that is going to be retained. So that means about 1.8 liters per day would be excreted. Well, the question becomes, why is this so stable? Why do we not have a continual, linear increase in GFR as we get to higher and higher hydrostatic pressures? Because this would reflect the hydrostatic pressure. And what we find is between this normal range of 80 to 180 millimeters mercury, there is a autoregulation that occurs to ensure that the filtration rate stays the same. So even though the hydrostatic pressure if we measure blood pressures or large arteries may be, say, at 120 or 180, the pressure that comes into the afferent arteriole is maintained constant. 3 ways this is accomplished. The first is what we call the myogenic response. This myogenic response refers to a stretch response found in the vascular smooth muscle in the arterioles in the glomerulus that respond to pressure changes. So as the pressure increases in the glomerulus, that smooth muscle is going to resist and respond to that pressure and either vasodilate or constrict as needed. The second control mechanism is something that we call the tubuloglomerular feedback loop. This feedback loop is under paracrine control, and we'll talk about it on the next slide. And that last mechanism is through both the autonomic nervous system and hormones. So the brain will utilize the autonomic neurons, the parasympathetic, or in this case, in the sympathetic neurons for our afferent arterioles and cause them to vasodilate or vasoconstrict as needed. We can also release hormones like angiotensin, renin that will get released. They can also alter the filtration coefficient that's occurring in those tissues. Let's look at the tubuloglomerular feedback mechanism that occurs. So on the afferent arteriole leading into the glomerulus, we have there are some cells that are associated both with the nephron and with the capillary, or with, in this case, the arteriole. It's called the macula densa. And in the macula densa, we have granular cells that act as sensors. So let's say that we increase blood pressure, and that causes an increase in GFR. So we start filtering a little bit more. That flow is going to start moving through the tubules. It'll flow past the macula densa. And in these cells, in the macula densa, they sense this increase in filtration, right? There's a stretch associated with that. And so they will start producing a paracrine chemical that then binds to the granular cells and causes the smooth muscle in the afferent arteriole to constrict. And as we constrict, what that's going to be is decreased blood flow into the glomerulus because the resistance gets increased, right? That decreases the hydrostatic pressure in the glomerulus, right? Why does it decrease it? Because we're building up pressure here, but we're limiting flow into here, so this would decrease the pressure on this side of our system. As that GFR decreases, then we have less flow through going through the tubules, and that would then restore the macula densa saying, okay, this is the proper GFR, we can stop producing this paracrine. So this is an ongoing process in which we are self-regulating the filtration that occurs within the nephron itself. So we've talked about filtration. Let's move on to reabsorption. So reabsorption, again, is going to be the movement of fluid or solutes from the lumen back into the blood. And it can be either active or passive. This can occur through transcellular transport, like we talked at the very beginning of the semester. So we'd actually have to diffuse through the epithelial cells of the lumen and then cross both what we call the apical and basolateral membranes. We'll see that in a second. We could have paracellular transport in which we move through the cell-to-cell gap junctions. And it can be mediated. So like in the case of in some cases, we have plasma proteins that are going to mediate what we call endocytosis, in which, again, we are moving substances like megalin back into our blood system. One of the few passive reabsorptions is going to be urea. So urea doesn't require any mediated transport. It'll passively be reabsorbed as needed. So let's look at a few of these transport mechanisms. Okay, so in the case of sodium is going to be reabsorbed by active transport. So it's going to be pumped against its concentration gradient as it moves. And this is going to be an example of, again, transcellular. So we're moving from the apical to the basolateral membrane through the actual epithelial cell. Anions, they can be moved either transcellular or paracellular to the gap junction. Same with water, same with potassium, calcium, or urea. Looking at sodium reabsorption, when sodium moves into the proximal tubule, it'll be actively transported and reabsorbed. We begin with having a high concentration of sodium, so we can move down its concentration gradient. So it'll move through the apical membrane using just an epithelial sodium channel. Okay, so we move down this channel. Now we have a lower concentration in here. Generally, there's a high concentration in the interstitial fluid. And so we're going to need to pump this out, and we'll use something like the sodium-potassium ATPase pump to pump sodium against its concentration gradient into the interstitial fluid to be reabsorbed into the blood. We can also utilize the sodium-linked reabsorption. This is secondary, or sometimes called indirect active transport. Again, where we use glucose and sodium together. So again, because sodium is high on that apical membrane, we can move it on its concentration gradient. We can utilize that to bring low concentrations of glucose into areas where there's a high concentration of glucose. So this is how we would reabsorb glucose. We use the secondary, that sodium glucose transporter. We then use the GLUT transporters to move from high to low, and again the sodium potassium pump to move it from low to high. Now, our renal transport can manifest a saturation point. So these figures from your book are looking at glucose. We see that primarily glucose filtration is going to be linear. So the amount of glucose that we filter is going to be dependent on the rate of that glucose coming into the Bowman's capsule. And it's going to be proportional to the plasma concentration. So the higher the glucose concentration, the more, and as we bring more in, we're going to be filtering more out. Or we don't want to lose glucose, so we have reabsorption of it, right? What we see is that we're able to reabsorb it until we hit our transport maximum. So at roughly 300 milligrams per 100 milliliter plasma, all of those GLUT transporters, so our secondary or sodium glucose transporters and the GLUT transporters are being utilized, and so at this point we're not able to reabsorb any more of our glucose. We're always going to get about 375 milligrams per minute. That's the transport maximum. Anything above this, so if we go to glucose concentrations above 300 milligrams per 100 milliliters, right, we're going to start hitting what we call renal threshold, in which we're going to lose glucose. Glucose is going to get excreted. So if you put all of these together into a composite graph, okay, we see where our transport maximum is. At that point, we would then have excretion of glucose. Secretion is going to be the movement of molecules from our blood or the extracellular fluid into the lumen of the nephron outside of the glomerulus. So this is an active transport process. And it's really important. Secretion is going to be important for both our potassium and our hydrogen concentration homeostatic regulation. We see this with our anion secretion, in which we would then secrete this sodium using the sodium-potassium pump. So we're bringing sodium out, but we then secrete potassium into the cell. And then that potassium can be utilized in the indirect active transport of additional organic anions into the cell. So in this case, we're bringing in dicarboxylate into the cell. That dicarboxylate then gets utilized to bring in the organic anions. And those organic ions will then get secreted into the lumen. Don't worry about all of these transporters. I'm not going to test over this. What I want you to know is that in secretion, it's generally an active process. We utilize either hydrogen or potassium, and what we're really usually secreting the most of is going to be organic anions. Our last process is excretion. So in excretion, we are looking at complete loss of a fluid or a solute from the body. So we could calculate excretion really by taking whatever's filtered minus what's reabsorbed plus whatever gets secreted, and that would equal our excretion. In physiology, we can calculate excretion rates utilizing a concept known as clearance. So clearance is the rate at which we would have a solute disappear from our body by excretion or by metabolism. So for example, if you were to ingest alcohol and we took a blood alcohol volume at a given time, we could look at how long does it take for that alcohol to either be metabolized or excreted from the system. And there's a couple what we call non-invasive ways by which we can measure this GFR. One of those is by ingesting a compound called inulin. Sometimes we'll also use creatinine. But inulin is a perfect GFR measurement tool because it's filtered and it's not reabsorbed and it's not secreted. So we know if we ingest we administer a set amount of inulin, Then as we watch those blood inulin levels decrease, we can infer what GFR is. Now that we've talked about these four processes, let's talk about how or where some common solutes are handled and where these processes take place in the nephron. So all filtration takes place in the Bowman's capsule. Everything is going to occur here in the Bowman's capsule where we have filtration out of the glomerulus. Now, as soon as we move into the proximal tubule, we have some reabsorption that begins. This is where we will start to reabsorb sodium, potassium, almost all of our glucose is going to be reabsorbed here. Okay, urea will be reabsorbed here. We'll also see in the proximal tubule that we'll have some secretion of an organic anion. Okay, so we have some organic ion secretion. Then we move into the descending limb. Really, there's not much that occurs in the descending limb as far as reabsorption, secretion, or excretion. But in the ascending limb, we do have some reabsorption, again, of sodium, chloride, potassium, and calcium, and secretion of urea. So here we'll have additional urea, if needed, can be secreted. The distal tubule, again, reabsorption just occurs here. We don't have any secretion. And we'll see sodium, chloride, and calcium being reabsorbed. Now, this is also one of the sites in which when we have hormonal control of reabsorption, this is where we can adjust like our sodium and our calcium reabsorptions. If we need to reabsorb more, it'll occur primarily in the distal tube. Finally, in the collecting duct, we will receive reabsorption of sodium, and then our final reabsorption of urea can occur here. We also see some reabsorption of potassium, and this is the area in which we can kind of regulate our potassium reabsorption is in this collecting duct. So if we have a normal potassium intake, then we will tend to excrete about 15% of our potassium that's found in the collecting duct. But if we have low potassium intake, then we'll have an increase in potassium absorption that occurs in the collecting duct. If we have a really high potassium intake, so we're loading up with potassium, then we can actually have, we won't have hardly any reabsorption of potassium here, and we'll excrete quite a bit. Okay, so make sure you understand this figure a little bit of which in the proximal tubule, distal tubule, ascending, and collecting ducts, what processes are going on. This table is also really good of kind of showing where we have regulated absorption. So sodium, the regulated absorption is going to occur in the distal tubule. For calcium, it's going to occur in the distal tubule. For our potassium, it's going to occur in the distal and collecting ducts is where we're going to have most of that kind of occurring. The last process that we need to speak about is micturition. So micturition is the physiological term for excretion and urination. And it's a unique process that utilizes both smooth and skeletal muscle. If we look at the bladder itself, it's made up of smooth muscle. And we have two sphincters. We have an internal sphincter that's smooth muscle, and there's an external sphincter that is comprised of skeletal muscle. Now, the skeletal muscle is contracted, that it's under tonic control, and so we have this contraction that's going. So pretty much all the time we have this, we have a somatic motor neuron that is being triggered to tell that external sphincter of the bladder to remain contracted. What happens as we start to fill the bladder is that stretch receptors found in the smooth muscle begin to discharge, and those stretch receptors send an efferent signal that then does two things. The first is that it's going to depolarize, so it's going to be excitatory for a parasympathetic reflex neuron that causes the smooth muscle in that internal sphincter to relax and the smooth muscle to start to contract. So we relax this internal sphincter through this parasympathetic neuron. Okay, the second thing is that it is going to inhibit the tonic discharge that is stimulating the motor neuron. So we have an excitatory pathway along the parasympathetic and an inhibitory pathway along the somatic motor neuron, which causes the external sphincter to relax. Now, all of us have experienced it before where we know our bladder is filled. We have that feeling of I have to go, I need to urinate, and yet we don't just necessarily wet ourselves, right? We don't urinate in our pants. Why is that? Well, because we have higher cognitive control from the brain that can override this inhibitory signal and maintain that external sphincter constriction, right? Now, where do we not see this? In small children, right? As they are developing and the brain is developing, early on they don't have this higher cognitive control. That's why a child is in diapers, right? It isn't able to override these stretch receptor senses. And so that external sphincter often is relaxed. As children age and begin to develop, their brains start to develop more and more, we reinforce this pathway, and they then are able to hold it, right? Now, in some children, right, they still have issues with this when they sleep. You can have the term, a bedwetter. And typically what happens is during sleep, in children that suffer from this, that cognitive pathway that's reinforcing this tonic discharge is lost during sleep, during deep sleep. And therefore, stretch receptors can override it and again lead to urination.